I was here saying that the problem of diabetes is that high levels of glucose over time affect many organs. So an epidemic of diabetes is in fact an epidemic of its complications. Now, large epidemiological studies have clearly shown that the complications of diabetes are linked to prolonged hyperglycemia. But the actual cellular and molecular mechanisms by which high levels of glucose affect the target organs of complications, still today, it's poorly understood. One reason for that is that there are no good animal models that recapitulate the complications of diabetes in the intensity and the combination seen in humans. So our lab over the past many years has established a link between the complement system and the complications of diabetes, published extensively, including a comprehensive review a couple of years ago in endocrine reviews. So briefly, the complement system is an array of proteins in the circulation that are the first line of defense against foreign bodies and bacteria. An activation of complement eventually forms a membrane attack complex on the membrane of the target cell, which allows the influx of salt and water, swelling and lysis of the target cell. Now, as you can see here, insertion of a MAC-pore into a red cell would induce swelling and lysis of the red cell. Also, insertion of MAC-pores into endothelial cells would induce the release of an array of growth factors and cytokines that together can induce proliferation, inflammation, and thrombosis, as characteristically seen in the target organs of diabetic complications. So it's understandable, having this system trying to poke holes into our cells, that an array of complement regulators, proteins, and other factors have evolved to restrict complement activation on self cells. CD59, shown here, is the most critical complement regulator in humans. It's a protein attached to the external surface of the cell through a GPI, glycosylphosphatidylinositol link, as shown here. And the only known function of human CD59 is inhibition of MAC formation. So the potentially harmful effects of complement on self cells is prevented by this delicate balance between activation and restriction of complement activity. If this balance is broken, there can generate very severe phenotypes. I'm showing here the brain of a 2-year-old offspring of a family with total deficiency of CD59 because of a nonsense mutation. And as you can see, at 2 years of age, this child has massive cerebral infarctions and retinal infarctions too. We have shown that in human diabetes, human CD59 is inactivated by glycation. And glycation is the non-enzymatic attachment of glucose to epsilon or alpha amino groups in proteins. In the case of CD59, an epsilon amino group. The glucose attaches, as shown here, and then rearranges to form this stable fructosamine called Amadori compound. Now, I mentioned before that glycation is a non-enzymatic process. But even though it's non-enzymatic, it has site specificity. Because in most proteins known to be glycated, there is a lysine residue very close, about 5 angstroms from the histidine, that provides acid-base catalysis for the reaction. In this case, as an example, in the beta chain of human hemoglobin, there are 3 lysines, 66, 65, and 61, very close by. But upon exposure to high glucose, only lysine 66 is glycated because of proximity to histidine 63. This then conforms what's known as a glycation motif, a histidine-lysine pair at about 5 or less angstroms distant. When the NMR structure of human CD59 was published, we found a putative glycation motif at the core of the active site of human CD59 that centers around tryptophan 40. The presence of a lysine 41 and a histidine 44 very close by suggested to us that this could be a glycation-prone motif in the active site of human CD59. To test this hypothesis, we purified and then made recombinant CD59, and we showed in a hemolytic assay that measures the activity of CD59 that indeed human CD59 upon exposure to high glucose in vitro, it's progressively inactivated as shown here. We went one step further and then mutated the histidine 44 and obtained a form of human CD59 that's active against human complement, but is not sensitive to glycation inactivation. And this was the first experimental demonstration of this concept of a glycation motif in a protein. Now, this experiment was done in very high glucose, 500 millimolar, because it's well known in the glycation research community that in vitro glycation for some reasons requires very, very high levels of glucose, totally non-physiological. Physiological glucose, as you know, is 5 millimolar. A poorly controlled individual with diabetes, 25 millimolar. So to answer the question of whether human CD59 could be glycated in vivo under more physiological conditions, over years of work, we developed finally an antibody that's highly, highly specific for lysine 41 glycated human CD59. And with this antibody, we show that in the urine of individuals with diabetes, there are high levels of glycated CD59 that's not present in the urine of non-diabetics. We also showed the presence of glycated CD59 colocalized as expected with activated complement proteins in the glomeruli of individuals with diabetes, not present in individuals without, and also in nerves, nerve biopsies from individuals with and without diabetes, and the small vessels inside the nerve, so-called vasa nervorum, express high levels of glycated C59 and the position of complement proteins. So now I will refer to the CD59, glycated CD59 as a biomarker in diabetes. As I mentioned before, CD59 is universally expressed in all cells. From there, it can detach and go into the circulation. And in the circulation, it can be found at levels that are about 150 to 200 nanograms per ml, and also in urine, 4 micrograms per ml. Hold on. Disconnect my phone. It's driving crazy. So over the years, we were able to develop antibodies against CD59 that allow us to develop an ELISA method to measure with high specificity and sensitivity glycated human CD59. A capture antibody that, as you can see, captures all CD59 in the sample, and a detection antibody that only recognizes the glycated form of CD59. We also were able to generate a synthetic calibrator to run thousands of samples, and this was also published years ago. So with this assay specific for glycated CD59, we were able to show in a first study of 400 individuals with and without diabetes that human CD59 is highly significantly increased in the blood of individuals with diabetes. In another study, we measured glycated CD59 in 109 individuals with no history of diabetes, who for the study underwent an oral glucose tolerance test, or OGTT. As you can see, glycated CD59 was a strong and independent predictor of the 2-hour plasma glucose in the OGTT. So based on this study, that glycated CD59 predicts the results of an OGTT, we asked the question, could glycated CD59 be a biomarker for gestational diabetes? Why? Because gestational diabetes, which is glucose intolerance that begins during pregnancy, is a condition today only diagnosed by oral glucose tolerance tests. And gestational diabetes is a major public health concern because it's a major risk factor for adverse pregnancy outcomes, from macrosomia, very big babies, or life for gestational age if they are below 4 kilograms, fetal dystocia, neonatal hypoglycemia, a series of very serious complications of pregnancy for the mother and the newborn. And importantly, I'm sorry about that, like gestational diabetes is a major risk factor for the future development of type 2 diabetes in the mother. For this reason, professional organizations around the world strongly recommend as standard of care screening for gestational diabetes all non-diabetic pregnant women. And the standard of care today is to screen, diagnose GDM at weeks 24 to 28 of pregnancy. This is done through oral glucose tolerance tests. In the US, mostly through a 2-step screening process, and in most other countries through a 1-step screening. So the 2-step screening consists of a first test. The pregnant woman drinks 50 grams of glucose, and there is one measurement 1 hour after the load. If that fails, meaning the glucose 1 hour after the load is higher than 140 milligrams, then the pregnant woman undergoes an OGTT that consists of 100 grams of glucose and 4 blood draws. And to diagnose GDM, there's need of 2 or more abnormal values. Needless to say that this test is uncomfortable, time-consuming, of low reproducibility, But importantly, both biomarkers currently used for diabetes control management, hemoglobin A1C and fructosamine, have no sensitivity whatsoever for GDM screening, and they're just not measured. So in our study, we conducted a prospective case control study of 1,000 pregnant women undergoing two-step screening for GDM. Of these, 500 had a normal and 500 an abnormal GCT. And as you can see, there were 55% of women with a normal OGTT, about 20% with one abnormal value, and 127 women with a formal diagnosis of GDM. Glycated CD59 was almost tenfold increased in the women who failed the GLT test, as you can see here. The test for GCD59 also totally separated both populations of controls and the women with formal diagnosis of GDM. With high sensitivity and specificity, as determined by an area under the curve, ROC curve of 0.92, and a positive predictive value of an odds ratio of almost 38 times. Furthermore, the prevalence of glycated CD59 in the mother was associated with a higher prevalence of large for gestational age babies. So with this in mind, we turn to early detection of gestational diabetes, which is a major issue because women developing GDM in the first trimester at a much higher risk of all the adverse outcomes and even more predisposed their offspring to diabetes, obesity, and cardiovascular disease. So we asked the question, could glycated CD59 serve to detect gestational diabetes earlier in the first trimester of pregnancy? So to answer the question, we partnered with a group of European researchers who had conducted the DALI study, a prospective multi-center RCT in 9 European countries. I'm sorry about this, sorry. So in the DALI study, participants were enrolled at week -20 in the first 20 weeks. Of 1,035 women, there were 237 with formal diagnosis of GDM. And this was expected, it was an enriched population to obtain more women BMI of 29. Of those, we received 693 samples, 486 of normal glucose tolerance, and 207 with early diagnosis of GDM. Glycated CD59 was significantly increased in the women diagnosed with gestational diabetes early in pregnancy, as you can see. The sensitivity and specificity was also quite high with the area under the curve of the ROC curve of 0.86. And interestingly, in this population, as expected, I mentioned before, hemoglobin A1C was absolutely no predictive value. Also, levels of glycated CD59 in the mother were associated with the prevalence of long for gestational age babies to the point that 1 unit increase of glycated CD59 was associated with a 30% higher odds ratio of a large for gestational age baby. It was also associated with other abnormal outcomes, as for example, a combined outcome of congenital malformation and neonatal hypoglycemia, 1 unit increase of glycated CD59 was associated with a 70% increase odds ratio for a combined abnormal outcome of congenital malformation and neonatal hypoglycemia. So Recently, the key opinion leaders around the world on gestational diabetes published in Nature Reviews this interesting comprehensive review of the subject and discussing early diagnosis and future biomarkers. They made this statement in the paper about the perspectives of plasma glycated CD59, the perspectives. So to conclude, plasma glycated CD59 seems to represent an accurate and patient-friendly test for screening and diagnosis, early detection, risk assessment, and monitoring of gestational diabetes. So, collaborators, grateful to the funding agencies that have supported this work for many years, and thank you to the audience for their attention, and I'm open for any questions you may have now. Thank you again.
Thank you so much, Doctor. Thank you for your presentation. Now the session is open for questions. If anyone had any questions for Dr. Josh Halperin, you can drop your questions in the chat box, or else you can unmute the audio and ask the questions for Dr. Josh Halperin. I repeat, if anyone had any questions, please drop your questions in the chat box. We have already left the mail ID of Dr. Josh Halperin so that you can reach him through the mail.